Okay, we're going to talk about bacterial shapes, as I mentioned in that introduction that I did on microscopy. And I'm gonna test you on some of these. I'm gonna have a little quiz, and it's not a quiz. It's basically going to be a little exercise for you to go through and see if you can figure out what the shapes are. Okay, it's going to be in the form of a quiz, but it's really an activity. All right, so first of all, let's look at the ubiquity of bacteria. It means they're everywhere. We've already talked about this. And they are very widely distributed in our soil, our air, our water. We have places that bacteria are normally found, areas on our body, in our body, and we call those normal flora. Those are microorganisms that live on or in our body that don't cause us harm. Okay. The only place where bacteria are not found are in areas that we purposely make sterile, as we talked about. Okay. Body fluids should be sterile. A fluid coming out of your spinal fluid should be sterile. Okay. We do a tap of your bladder, that urine should be sterile. Blood should not have organisms in it. Okay. We will work with clean rooms where we make sure that they may not be totally sterile, but the bacterial load is down as far as we can get it. Or we actually do sterilize equipment, surgical equipment, things that we're using. We autoclave. It's a special sterilization technique that we use in the labs and in the surgical sterile prep rooms. You autoclave instruments, high heat with water, and it sterilizes these instruments. Or in the lab, we'll take these plates that we grow these organisms out with, and we sterilize them, we autoclave them after we use them, and then we can throw them in the regular trash because we don't want these organisms to get out where it makes you guys sick. Okay? All right. So again, what makes an organism a bacteria? We've kind of gone through this. It's defined by their cell structure, their size. We talked about they lack a defined nucleus. They have a membrane. They have a cell wall that contains peptidoglycan. They don't have organelles like these membrane-bound organelles. They do have ribosomes, and they're small. And I've got a picture here of a red blood cell, which is about 6 to 8 micrometers. Sorry about there, microns. And you can see the size of that red blood cell right here, white blood cell, a little bit bigger. Can you see that little dot? That's the bacteria. There's a little tiny bacteria. See the two little guys together right there? Okay. And if you gram stain this, you can see the little cheek cells and you can see the little bacteria. So these guys are, again, little guys. All right, when we handle these, we need to use aseptic technique. Hopefully that and you've learned some of this in the little YouTubes that I've given you. But this is where you keep the unwanted microorganisms out. Okay? This is not sterile. This does not mean sterile. This means that we've reduced the numbers of the bacteria. So when you disinfect a working station, not all the bacteria are going to die. But the idea is to get that microbacterial load down, really low. Okay? There are times when we do need to be sterile, and that's when we, as you've seen, we use a flame to sterilize a loop or a needle to sterilize it. Or, as we mentioned, we can autoclave plates and tools and tubes and stuff like that to kill the bacteria. Obviously, we can't put anything plastic in there and anything like that, but we can do that kind of stuff. We keep our cultures covered at all times until we're ready to use them, and then you have to be aware if you're actually in the lab of your area. If you're, you need to make sure that you're not contaminating your area. Okay. As we mentioned, these organisms like primarily to divide by binary fission. And so he's a little lonely guy, but not dividing. So you inoculate this agar plate, and this is an agar plate, and you put it in the incubator, and overnight it comes out. And you got this. And you can see this is a streak, and you can't get an individual colony. We'll talk about that. And here's an individual colony here and here. And each colony has millions of bacteria in it. Millions, okay? And what we try and do with streak plates that we'll talk about is get it so we can pick out an individual little colony so we can work with it, because you can contaminate if the culture is not a complete culture of the same organism. You want to pick up what you want to pick up, okay? But this allows us, there's millions of guys in this little colony, and that's what we're looking at when we grow them out on a plate. Now, yeah, you see, try and pick out an individual colony. Yes, I can, because I am good at it. Like that guy there is all by itself. But when you have them touching, that's what I'm talking about. It's hard to go in and get this one little organism when everything else is around it, and you want to try and separate it out. So when you look at these plates, you're looking at the morphology of the colony. What does the surface look like? How high is it? Notice this is higher, this is more mucoid, this is flat, and this is dark. This has a different color. So the color and the edges and the elevation can lead us to it being a totally different organism. We realize it's different. Okay, and so when we look at a plate, we may have one or two different organisms in these plates. We get a specimen down, it may be full of 2 or 3 different organisms, or if it's a throat, it's like the entire organisms that's in a throat. We're looking at one particular thing. It has a smell. And when you open it, there are some very characteristic smells. I will tell you, the microbiology lab probably is the worst smelling place in the entire laboratory because of the smells of these organisms and the specimens that we get. And you just have to get used to it. And then we look at the morphology. Okay, the size of the organism, the shape of the organism, the gram-stain reaction, gram-positive cocci and clusters, gram-negative bacilli. You're going to start to hear me say this so that you guys get an idea when you look at this. So when we look at the shapes, we've already kind of gone over this a little bit, but bacillus, singular, bacilli, plural. Rod-shaped, and you can have very short bacilli. This is Pseudomonas aeruginosa, famous for ear infections and lung infections. Can get pretty nasty. You can have long bacilli, which is bacillus, or you can have a particular kind of bacillus, a different bacillus within the spores. And you see those little white spaces? See those little white spaces? Those are the endospores. Okay, so it's actively producing endospore, and not in a happy mood right now. All right, cocci, spherical. We talked about that, and it can be depending upon whether it's dividing in how many planes, whether it's going to be chains or clusters. Remember, our bacilli only divide in one plane. So you're not going to cluster a bacilli. Can you see these all little separates and maybe a little chain or something like that? But you're not going to get clusters. Cocci can cluster. The Staphylococci cluster. So, and we'll talk about that. You can see that there it is singly, in pairs. There's particular tetrad. It's a real good one. A little bit more. You can see a little, that's a tetrad, but it's on its edge. But you can see this kind of like grape-like clusters. That's notorious for staph. So that's reported out as cocci clusters. And when you get a full gram stain report, if you start to read them, can you see them? It will say gram-positive cocci, singly, in pairs, and clusters. That tells, again, that doctor something. Gram-positive cocci in chains. That tells them it's not staph, it's a strep. Okay, so here's your little strep. Do you see that long chain? Okay, so this is divided in one plane. This is divided in more than one plane. Okay. All right. Cocci are classified singly, pairs, tetrads. If they remain in pairs, if you just see pairs, they're reported as diplococci. And again, if they continue to divide along and form a chain, it's streptococci. Okay. And you can see on and on. So this is the division of cocci. You can see how they look. There's your strep, there's your tetrad, there's your cluster. Okay? And you can see the difference. Okay, diplococci and streptococci. You can see there's a diplococci right there, and right there, and right there. But you have chains. This particular smear, that's all we see are diplococci. There are no chains. And that is the difference what a microbiologist sees when they look underneath the scope. We look at what we're seeing. I'm looking at this, and all I see are diplococci. I look at this, and I see diplococci. I see chains. Oops. So it would be streptococci that we would report out. Okay. So cocci in chains is how it would be reported out. And then you can see the tetrads. I specifically picked out one. You can see the 4 and 4 and 4. Okay. And there's your groups of 8. There are particular organisms that do in 8. Your genera. Okay. Your staphylococci, again, notice pairs. And here's a better one. Okay. Singly, in pairs, there's a tetrad right there. Okay. There's little, there's a 6. There's arsine there. But here, see how it's clustered together? That's a cluster. Okay. Vibrio. These are curved rods. They are not all straight. Okay, if you look at that, you see that I've got it pointed out. You see that curve there? The curves. See the curves? This Vibrio cholerae causes cholera. This is notorious. This is what we look for. Okay, it's comma shaped. All right, so these are just the basic shapes. This is real short and real sweet, but I wanted to do another basic one for you guys so that you can really understand. And again, I want you to, I'm going to have different, go to the little quiz. It's not a quiz. It's not counted as a quiz. It's counted as an activity. And I'm going to be having different pictures. Can you identify? Do you understand these different shapes? Okay? All righty. And that's it.